Hello, everybody. Good evening. Today, for our Cell Talk presentation, we have Dr. Vijay Padul, who is a senior scientist at Renix and who leads the bioinformatics, genomics, and the next generation sequencing research related activities at Renix. Currently, he is working on the new antigen cancer vaccine pipeline projects. He has a PhD in life sciences from TMC, Actrec, Homi Bhabha National Institute, Navi Mumbai, and his work during his PhD focused on the cancer genomics. Previously, has also worked as assistant professor at undergraduate and postgraduate institutions. At Renix, he has gained experience in scientific, operational, as well as administrative activities. Over to you, Dr. Vijay.
Thank you, Dr. Divya, for the introduction. So for today's Cell Talk Journal Club, I have chosen this article, Molecular Landscape of IDH Mutant Astrocytoma and Oligodendroglioma, Grade 2, indicate tumor purity as an underlying genomic factor. The original authors are Bingao Zhao et al. The topic selected for this presentation is astrocytoma. The journal in which this paper has been published is Molecular Medicine. It has an impact factor of 6.159. It was published in year 2022. And this research presented in this research article has an impact on grade 2 glioma subtype diagnosis and prognostication. Introduction. So this research is focusing on the molecular differences between astrocytoma and oligodendroglioma that are the grade 2 subtypes. Now, astrocytoma and oligodendroglioma itself are the histological subtypes of the glioma brain tumors. So, molecular features among broad astrocytoma and oligodendroglioma subtypes, grades, that is grade 2, 3, are known and it has been used for the classification of gliomas based on molecular-based classification, and which has been recommended by World Health Organization classification of tumors of the central nervous system, and it has been recommended as an ideal classifier for the glioma subtypes. But still, even if we have the molecular characteristics such as IDH mutations and 1p90q co-deletion to stratify the glioma in such a way like IDH mutations and 1p19q co-deletion, if it is present, then it is one subtype which is mostly enriched with oligodendroglioma histology, and IDH mutations without 1p19q deletion as an astrocytoma subtype. But even in this case, what are the molecular basis or what is the molecular reasons for the histological differences between the astrocytoma histological subtype and the oligodendroglioma histological subtype? That is not known. So this research article is attempting to identify those differences between the histological subtypes of glioma tumors. Now, along with the recommendation by the classification system also, and there are several studies which have underlined the superiority of molecular features over the histological features for the classification. But still, for scientific reason, it is interesting to know what are the molecular differences that lead to the histological differences between astrocytoma and oligodendroglioma tumors. So here in this research articles, they have considered on the tumor purity, and tumor purity is estimated as a measure of proportion of tumor cells in the tumor along with normal cells. So in a high purity tumor, the tumor cells will be uniformly distributed across the tumor section, the histological section that is studied under microscope. And if there are multiple different types of cells or different morphological types of cells are observed, then that tumor is supposed to have low tumor purity. So low-grade tumors tend to have high tumor purity as compared to the high-grade tumors, because as the tumor grade increases, the cell becomes more aggressive and they diffuse in the surrounding area, and also due to their genomic alterations, they tend to have different morphologies also. Along with that, there are different other immune cells, fibroblast cells, endothelial cells. These are present in the tumors, and that also contribute to the definition of the tumor purity of the tumor section. Now, the tumor purity is routinely estimated by the pathologist by analysis of histopathological sections of the tumors, and this can also be done by image analysis by employing the computational methods, where image taken high resolution image that is taken by using a microscope, it is fed into an algorithm, and it will identify the tumor purity and give a specific value associated with the tumor purity. So lower tumor purity means higher grade, and higher tumor purity will define as the low grade of the tumor. And if the tumor purity is low, that means heterogeneity is high. That means this tumor is behaving in an aggressive manner, and it is diffusing into the surrounding areas, and that will lead to the classification as a diffuse glioma. And as aggressive nature of the tumor increases, it has shown that it leads to the failure of the treatment, the routine chemotherapy and surgical resection treatments. These are highly associated with the low-grade tumor, and success is mostly dependent upon the resection of complete tumor, and that is possible mostly with the high purity tumors. A general background about this paper. So here the authors have attempted to investigate by using multi-omics approach and the clinical data of the only grade 2 IDH mutant astrocytoma and oligodendroglioma histological subtypes to identify what are the molecular differences between these two histological subtypes of the tumors. Now, the present challenge that is leading to this research is that molecular differences between grade 2 mutant astrocytoma and oligodendroglioma histological subtypes, that is leading to the histological subtypes, is not known. And here the authors are attempting to identify that. So objective of the research was grade 2 IDH mutant astrocytoma oligodendroglioma tumors have an indolent natural history, and they are recognized as a distinct entities of neoplasm, but there is little knowledge about on the molecular differences between them. So grade 2 itself is they are very indolent. That means they take time to grow and infiltrate the surrounding area. But even if in such cases, what are the molecular aspects that are leading to this behavior, that is not known. And this study attempts to explore the molecular differences between grade 2 IDH mutant astrocytoma and oligodendroglioma. Methodology. So they have done multi-omic analysis. So that included mRNA expression, somatic mutation, copy number alteration analysis, then DNA methylation analysis, microRNA signature analysis, epigenetics data analysis, and in the proteomics aspect, reverse phase protein arrays, that is RPPA data, it was analyzed, and patient demographic information such as clinical information, grade, gender, etc. So they did this multi-omic data analysis on two data sources. First is the Cancer Genome Atlas Glioma project that makes available this data online, and another one was Chinese Glioma Genome Atlas project. So in this methodology, they performed different analysis with different aims. So somatic mutation and copy number alteration analysis was performed to identify what are the different genes that are mutated and what are the genes that are either amplified or deleted due to copy number deletions or amplifications in those different patient sample types. Then biological functional analysis included what are the functions of first, what are the genes that are enriched in the mutation and copy number alterations, and what are the biological functions of those particular genes. Then evaluation of DNA damage repair signature and tumor purity. So DNA damage repair genes they play important role in the tumor biology. If these DNA damage repair genes, if they are not functional or adequately functional or mutated, then one more avenue for controlling the tumor is lost. So it is important to identify whether DNA damage repair related genes are intact in the tumor or not, and its correlation with the tumor purity. Then next, they identified glioma cluster profiling. So there are different research articles that have been published throughout the years. They have attempted to identify different clusters of glioma samples by using different statistical and computational approaches, by using mRNA expression data, microRNA expression data, copy number data, etc. So they also did this glioma cluster profiling on these two data sources. Then they attempted immune microenvironment quantification. So as it has becoming apparent that immune suppression is also an important aspect in the tumor survival, tumor cell survival and growth. If they can evade the immune system, then the tumor will grow. If immune system is able to detect it, then there might be a possibility of clearing of that tumor cells. So how much is the immune microenvironment is enriched in this tumor types, it was identified. Then they use machine learning pipelines for astrocytoma and oligodendroglioma gene panel identification. Here they have attempted to identify certain gene signatures which will be able to identify histological subtypes astrocytoma and histological subtype oligodendroglioma as different from each other. So coming to the results, so this result shows the somatic mutation landscape of the tumor subtypes astrocytoma and oligodendroglioma. So the first figure, it shows the combined mutational signature of low-grade gliomas that combines astrocytoma and oligodendroglioma, and the other two are individually showing astrocytoma and oligodendroglioma. So as can be seen in low-grade glioma, IDH1 mutation, TP3 mutation, ATRX mutation, CIC mutation, and TT10 mutation, which are found to be more prominent. These were also found to be prominent in this combined database also. Then when we separate histologically astrocytoma and oligodendroglioma, naturally there is IDH1 mutation is present in all the samples, but in astrocytomas, T53 mutation percentage is more as compared to the oligodendroglioma, in which it is very low. While in oligodendroglioma, CIC mutation and PIK3CA mutation is more enriched than as compared to the astrocytoma. If these tumors were stratified according to the molecular signatures, then it will be 100% separation between IDH mutation and TP3 mutation on one side, while IDH mutation and CIC mutation on another side. But here, the separation or the classification is based on the histology. So therefore, even if it is astrocytoma histology, molecular oligodendroglioma might be present here, and therefore certain CIC mutations will be present here. And as this is oligodendroglioma, still molecular astrocytoma features will also be present in these cases, such as ATRX mutation and TP53 mutation. Now here, this result shows the somatic mutation landscape with respect to the mutation frequency of genes in key pathways. So here, these are different key pathways labeled by different colors. So this is chromatin modifier genes, then DNA damage repair pathways, TP3 pathway, RB pathway, retinoblastoma pathway, and their corresponding mutation frequencies. So as you can see, there in DNA damage repair pathway, these are more enriched in astrocytomas as compared to oligodendrogliomas. That means these mutations are present in more number in astrocytomas as compared to the oligodendrogliomas. Then similar differences were identified with respect to TP3 mutation and retinoblastoma mutations also. So this is just to identify when we subtype this histologically, whether there are certain pathways that get enriched with respect to mutation or not. So this main difference is with respect to the chromatin modifier genes. So here in astrocytoma, ATRX is highly mutated, so it is enriching in this pathway subtype as compared to the oligodendroglioma, in which its percentage is very low. So in this way, they identified the somatic mutation landscape with respect to the key pathways. The next, they also subdivided this entire data set and its mutations into 4 subgroups. These 4 subgroups were defined as ApoBEC, unknown, CT2, CPG, and the last one. Here they have found that in all the 4 subgroups, the transition from C to T, cytosine to thymine, it was more enriched. So this is the cosmic mutation signatures, and they have identified this particular pattern. The next, they studied tumor mutational burden, and they have found that the tumor mutational burden, there is not much significant difference with that respect in astrocytoma and oligodendroglioma. As this being the low grade, if it had been higher grade, naturally astrocytoma would have more number of mutations as compared to the oligodendroglioma. but these are grade 2 mutations. Then somatic mutation proportion of the well-known genes that are mutated in the glioma. So as you can see the differences with respect to ATRX, TERT, TP53, PTEN, and PIK3CA. So histological astrocytoma, it has a more percentage of ATRX mutation as compared to the histological oligodendroglioma. So 78% to only 1%. Then with respect to TERT mutations, it is more or less same, 199%. TP53 mutations are more present in astrocytoma as compared to the oligodendroglioma. PTEN mutation status is similar. Then PIK3 status is also somewhat similar, only like in oligodendroglioma, 2.3% as compared to the 2% in the astrocytoma. Then next, they performed the copy number alteration analysis and DNA damage repair related gene signatures. So here this shows chromosome 1 to chromosome 22, and these blue segments, they represent the deletion, and red they represent the amplification. So 1p and 19q co-deletion, it is the characteristics of oligodendroglioma, and it is seem to be enriched in the oligodendroglioma histological subtype, while chromosome 7 amplification, it is the characteristics of astrocytoma, so it is enriched in the astrocytoma histological subtype. And also they have seen the status of different genes such as CDKN2A, 2B, PTEN, and RB1. So its deletion was found to be prominent in the astrocytoma subtype. The next, they performed copy number alteration analysis and DNA damage repair related signatures. And here they are showing the proportion of amplification and deletion of key genes or the driver genes in the glioma. So ATRX, TERT, TP53, PTEN, CDKN2A, B, PDGFRA, RB1, and MDM4. So well-known differences, they were also present in this panel also. So astrocytoma and oligodendroglioma differ with respect to ATRX. They differ with respect to TERT mutation also, TP3 mutation also, and MDM4. And there are minor differences with respect to RB1, PTEN, and CDKN2. Then they also probed copy number alteration frequency in different genes that are associated with the glioma biology. So here the red represent amplification and green deletion, and this is the copy number alteration frequency with respect to that particular gene. So BRAF, EGFR, MDM4. So these in both this entire genomic panel, these were seen to have these genes were seen to have more amplifications as compared to in this category of genes where there were more deletions, PTEN deletion, RB1 deletion, CDKN2 deletion, CDKN2 deletion, whereas amplifications of these genes, EGFR, BRAF, etc. Next, they also found a significant difference between the number of copy number alterations observed. So number of copy numbers were significantly higher in astrocytoma as compared to oligodendroglioma. Microsatellite instability, that is also a measure of more genomic perturbations. So these were found to be not much different in the both histological subtypes. Then here they represent the status of different molecular alterations with respect to histological astrocytoma and oligodendroglioma. So here a copy number alteration burden was found to be more in astrocytoma as compared to oligodendroglioma, and this upper panel shows its actual distribution, and here it shows the statistical analysis which shows us the summary of how this entire glioma panel is having that particular status. Then aneuploidy score, tumor purity, then ploidy score, subclonal genome fraction, loss of heterozygosity, homologous recombination deficiency, PARP, isomer sensitivity, and TP53 score. So they found significant differences in this with respect to these different genomic alterations in the histological astrocytoma and oligodendroglioma subtypes. Then they identified the correlation between DNA damage response related pathways and tumor purity. So here in this panel, these are the different DNA damage response related pathways, that is base excision repair, nucleotide excision repair, then non-homologous end rejoining, direct repair, etc. So they have found that there is a correlation between the tumor purity in astrocytoma and oligodendroglioma. So green means that there is a higher correlation, and red means there is a low correlation. So they have found that there is a higher correlation with respect to certain pathways with tumor purity in astrocytoma and also in oligodendroglioma. So here they perform the prognostic significance of this different molecular alterations and its impact on the patient survival. So these are the different survival plots. So here it shows overall all patient survival plots. So here the green is oligodendroglioma and yellow is astrocytoma. So as it is well known, oligodendroglioma patients tend to survive longer as compared to astrocytoma, and it is becoming apparent in this combined panel also. So looking at other characteristics, MGMT promoter methylation, if it is present, then the oligodendroglioma, those oligodendroglioma patients will survive longer as compared to astrocytoma. There is also a difference between ATRX wild type status patients, then TERT wild type patient status, oligodendroglioma is surviving. Then here MGMT promoter methylation subtype patients, ATRX wild type patients, and TERT wild type patients. In these also oligodendroglioma patients are surviving as more as compared to the astrocytoma patients. So here this is the overall survival and this is the progression-free survival, and here they have subdivided each patient class into molecular subtypes based on chromosome 7 and chromosome 10 amplification and deletion respectively. So if chromosome amplification is present, if it is absent, and if chromosome 10 deletion is present and if it is absent, if we subtype in those 4 subgroups, what will be the survival status? So they didn't identify any significant in these 2 groups, but in the last 2 groups, ATRX wild type and TERT wild type patients, they have found that chromosome 7 and chromosome 4, its presence makes a difference into the survival of the patients. So that is also significant molecular characteristics that differentiates between the histological subtypes of the astrocytoma and oligodendroglioma. Then coming to the landscape of the multi-omics characteristics. So by combining all these molecular characteristics, what is the picture that is coming up? So they have presented it here. So so IDH mutant astrocytoma grade 2 and IDH mutant oligodendroglioma grade 2. So here the first panel, it shows the mRNA SI, that means mRNA expression based stemness classifier. So this is a statistical measure that identifies the stemness of this glioma subtype. So here it shows its proportion, and then in the second panel it shows the clinical characteristics of that particular patient, such as gender, then RNA-seq clusters, and then TCGA subtype, histology, etc. Then in the next panel, it shows the correlation of the purity and the DNA damage repair pathways. So here, loss of heterozygosity, purity, subgenomic fraction, and T53 scores. These different scores were calculated statistically, and its correlation with the tumor purity was analyzed. Then mutational signatures, like blue means low number of mutations and yellow means high number of mutations. So that is presented here to identify like which subtype will have a higher number of mutations. And then metabolism signatures. So again, red means higher metabolic activity and blue means a lower metabolic activity, and that with respect to citric acid cycle, urea cycle, and methionine cycle. it was analyzed. In the next part, they uh, explored whether there are any pathway-specific differences between the astrocytoma and oligodendroglioma histological subtypes. So here, this panel shows the relative expression of different genes in astrocytoma and oligodendroglioma. If it has red outline, then it is astrocytoma. If it has blue outline, it is an oligodendroglioma. And they have found some statistical significance in the expression pattern in TP53 pathway genes with respect to TP53, MDM2, MDM4, which is not significant, and PPM11D. For RB pathway, also they have found high significance with CDK and 2B and RB1. In RTK PI3K pathway, they have found significance with respect to the expression of PDGFRA, then RBB2, NF1. Now, in this analysis, they have identified, they came across number of mutations in a gene that is PTPN11. Now, this PTPN11, it is a phosphatase enzyme. It encodes a phosphatase enzyme which negatively regulates the PDGFR and EGFR, which are oncogenes. So PDGFRA and EGFR, which are kinases, which phosphorylate the downstream pathway genes and activate them and lead to the oncogenic pathways. So PTPN, its function is in the reverse direction. That means it will reverse the added phosphate and remove it from the protein structure and thus inactivate those particular downstream genes. So they have identified this pathway which leads to the dual regulation of EGFR and PGFRA, which is mediated by the PTPN11. And they have identified those particular regions where these phosphorylations are taking place. So this is tyrosine 62, tyrosine 546, and tyrosine 584. Then next, they studied the immune infiltration of the tumor samples and checkpoint immunotherapy response in the tumors. So here, they calculated immune cell infiltration by using different software approaches. The different softwares are available, which will take the data and it will identify how much of the immune cells infiltration is present in that particular tumor types. So it is based on the immune cell signatures. So they have identified those signatures in astrocytoma and oligodendroglioma. Again, the yellow being the higher and blue being at the lower side. So as you can see, most of the immune infiltration is more enriched in the astrocytoma as compared to the oligodendrogliomas, and this has been established by using multiple software approaches. Then they also analyzed the tumor microenvironment by using one method that is called as IOBR method. So here also they find the difference in the astrocytoma and oligodendroglioma, in that in astrocytoma there is a higher tumor microenvironment signature as compared to the oligodendroglioma. And it is known that oligodendroglioma respond less to immune checkpoint immunotherapy as compared to astrocytoma, and that is due to the immunologically cold nature of oligodendroglioma tumors. That means immune infiltration of the immune cells is not present in the oligodendroglioma tumors as compared to the astrocytoma, and that is also a factor why oligodendroglioma may have the higher tumor purity as compared to the astrocytoma. So that is becoming apparent in this particular analysis. Next, they perform gene set enrichment analysis in astrocytoma as compared to the oligodendroglioma. So here, this upper genes, gene sets, they are more enriched in astrocytoma. And if you can see, these gene sets are inflammatory response genes, allograft rejections gene sets, interferon gamma response gene sets, and epithelial mesenchymal transition gene sets. So these gene sets, these are mostly related to the immune environment of the immune system or immune environment of the tumor, and these were found to be more enriched in astrocytoma as compared to the oligodendroglioma. So when they checked what is enriched in oligodendroglioma, then they see that myogenesis and KRAS signaling pathway related genes were more expressed in or more enriched in the oligodendroglioma. So they have established this difference in the astrocytoma histological subtype and oligodendroglioma histological subtype with respect to the immune infiltration landscape. The next, estimate and absolute, these are algorithms which are used to establish the tumor microenvironment and immune system infiltration related scores. So these are the different scores, that is immune score, inflammation score, stromal score, tumor microenvironment score, and tumor index. So they found there is significant difference with histological astrocytoma and histological oligodendroglioma grade 2 with all of these aspects. Then next, they found the associations between tumor purity. All these panels, tumor purity is on the y-axis, while immune cytolytic activity, exhausted CD8 plus T cells, T cell inflamed expression profile, immune checkpoint profile, and myeloid-derived suppressor cells, all these related to the immune infiltration. So what is correlation with the tumor purity? So they have identified a negative correlation. That means if higher is the tumor purity, then there is a reduced or low level of immune infiltration signatures are identified in the tumors. And this is more prominent with respect to the oligodendroglioma. As you can see that most of the blue points represented in oligodendroglioma are more towards the high purity side. Then here they studied the different scores that are related to the immune checkpoint immunotherapy response. So TIDE score, that is a measure of cytotoxic T lymphocyte infiltration, and then the immune phenotype score, then PD-L1 expression, T cell exclusion. That means if T cells are not present in the tumor cells. So there they have identified statistically significant difference in the astrocytoma and oligodendroglioma subtypes with respect to all these scores. And here they have shown that oligodendroglioma T cell exclusion is statistically significant as compared to the astrocytoma. Then the last panel, it shows how the immune checkpoint therapy responders molecular features are behaving. So here, if astrocytoma is considered, then 56.4% of these patients are responders as compared to the only 25% of the oligodendroglioma histological subtype. So this is the difference that oligodendroglioma tend to have low response to the immune checkpoint immunotherapy. So this difference they have identified in this analysis also. The next, by identifying all this multi-omic analysis, everything that they have done, so they selected number of different predictors for diagnosing the histological subtype astrocytoma and oligodendroglioma, and also an important factor, that is tumor purity. And they have proposed a diagnostic model, and they propose that this is a superior model, and it will improve the patient stratification and also prognostication. So they have identified a difference in the tumor purity in astrocytoma and oligodendroglioma. But when the tumor samples are subclassified based on chromosomal alterations, based on chromosome 7 amplification and chromosome 10 loss, so all those four combinations, chromosome 7 is not amplified, but chromosome 10 is lost, chromosome 10 amplified, chromosome 10 lost, chromosome 10 amplified, but chromosome 7 amplified, but chromosome 10 is not lost, and both chromosome 7 is not amplified and chromosome 10 is not lost. So what is the several difference? So here with respect to the tumor purity, so they did not identify any difference among these subgroups when tumor purity is considered for astrocytoma, but when oligodendroglioma samples were considered, they have identified significant difference. So significant difference is seen in chromosome 7 gain and chromosome 4 loss subtype. So this is also an important prognostic criteria. In the next analysis, they performed gene set variation analysis, and they did it with high tumor purity samples and low tumor purity samples. in oligodendrogliomas and astrocytomas. So here, these blue gene sets, these are significantly enriched in astrocytomas, and the gene sets that are represented in green, these are significantly enriched in the oligodendrogliomas. And they have identified what is the variation when tumor purity is considered, when high purity and low purity tumors are considered. So they did identify some differences with respect to the tumor purity in the gene set variation analysis. So when they performed this analysis with high purity and low purity subgroup of these tumors, they identified 111 prognosis related differentially expressed genes, and they identified it by using a random forest model. So by using this 111 genes, they proposed a astrocytoma oligodendroglioma panel. So in order to study that, they created confusion matrix analysis by using different statistical methods such as ADAPT-boost, GBDT, XGBoost, etc. And all of these three methods performed very well. So this is a positive predictive value and negative predictive value. So positive predictive value was 100% in XGBoost and ADAPT-boost, while negative predictive value was 96% and 96.4% equally, while 100% in the case of XGBoost. So these statistical models that they created, it performed well, and they gave excellent predictive accuracy for distinguishing between the astrocytoma and oligodendroglioma histological subtypes by using all these molecular signatures. So this panel shows how that those molecular signatures are classifying the histological subtypes. So here, this astrocytoma oligodendroglioma panel classifier that was constructed by using a number of different genes and molecular alteration signatures. So they showed significant statistical difference with respect to astrocytoma and oligodendroglioma. So they created a classifier index, and if that classifier index value is less than 0.9767, then that is more likely to be oligodendroglioma. And if this classifier index is more than 0.9767, then it is more likely to be astrocytoma. And out of those 111 genes, they identified these 6 highly significant genes: HS3ST1, CNN3, SLAIN1, ABTB2, TRIM67, and DRG2. So as you can see, by using these classifiers in TCGA cohort as well as CGGA cohort, in both this cohort, this classifier model was able to subgroup the tumors in the histological astrocytoma and histological oligodendroglioma subtypes. So these are the different molecular factors also that are present, and tumor purity is also an important factor, along with the already known different genes such as ATRX mutation, TERT promoter mutation, MGMT methylation, etc. So in conclusion, with all these results, so this study has comprehensively analyzed by using 2 different data sets, the glioma histological subtypes, oligodendroglioma and astrocytoma. And by this multidimensional analysis, we can see that there are differences with respect to the molecular features, even microenvironment, tumor purity, and classification predictors, and also prognosis in these 2 histological subtypes. Next, if continued with this computational analysis, these results will facilitate the deeper understanding of diffuse glioma heterogeneity and tumor microenvironment interactions, and these findings will help to improve the patient's clinical benefits. Coming to the impact of the research, the study facilitates a deeper understanding of molecular features, human microenvironment, tumor purity, prognosis in grade 2 IDH mutant astrocytoma and oligodendroglioma histological subtypes. Then next, a study identified tumor purity as an important genomic factor, and which closely correlates with the DNA damage, repair signature, and copy number alterations. Finally, the authors claim that the diagnostic tool that they have developed by using machine learning approach and different molecular signatures, it will support the clinical decision making, and it will help to improve the clinical benefits for the patients. That's it. Thank you.
Thank you, Vijay, for this enlightening presentation. I don't see any questions from the audience. Yeah, I have Nopur. She has asked a question that they found 111 genes from machine learning. How do they select 6 genes from the panel of 111 genes?
So they did perform more analysis on that 111 genes, and they selected the topmost 6 genes which had more ability to differentiate between the astrocytoma and histological oligodendroglioma subtypes. So more details are there in the paper, but these are different statistical methods and algorithms.
So I hope this clarified your doubt, Nopur. I don't see any more questions here, so I thank you once again. Can we move on to the last slide, Vijay? Yeah, the next presentation will be by Jay Darji, and the topic is a hybrid CNN GLCN classifier for the detection and grade classification of brain tumor on 23rd February. So thank you everybody present here. Hope to see you on the next presentation.